Understanding PCR, a DNA amplification technique. PCR, Polymerase Chain Reaction, is a lab technique used to make many copies of a specific DNA segment. It's like a photocopier for DNA, helping scientists study genes, detect diseases, and even solve crimes. Steps of PCR. PCR happens in 3 main steps, repeated in cycles to create millions of DNA copies. 1. Denaturation. Heat breaks DNA apart. The DNA is heated to 95°C to break it into 2 single strands. Think of it like unzipping a zipper. 2. Annealing. Primers attached to DNA. The mixture is cooled to 50,065°C, so primers, small DNA pieces, can attach to the target DNA. Primers tell where copying should start. Through extension, DNA copies itself. The temperature is raised to 70°C, and an enzyme called Taq polymerase starts adding new DNA bases. This creates 2 identical DNA copies from the original. Final result. After repeating these steps 30-40 times, millions of copies of the target DNA are made. Where is PCR used? Forensics helps in solving crimes with DNA evidence. Medical diagnosis detects viruses like COVID-19. Genetic research studies genes and diseases. PCR is a powerful tool that helps scientists in many ways. If you found this interesting, like and subscribe to IdeaZoom Academy for more amazing videos.